Hello, and today we are going to review Chapter 5 called The Working Cell. And what this is going to allow us to do is to take a look at how the cell, the various functions of the cell, other than, since we have now covered all of the structures of the cell, we're going to look and see how the cell functions. We're going to basically focus on the plasma membrane, and then we're going to focus on the way cells get substances inside and way they remove substances, so sort of crossing that membrane. We're also going to talk about cellular energy in the form of ATP, and we're going to talk about enzymes. So it's it's a busy chapter. There's a lot going on, and this might take a little bit of time to get through, but we'll get there. So remembering from chapter 3, when we talked about the molecules of cells, we have these phospholipids, and here the phospholipid is going to be the gray oval with the two yellow fatty acid tails. And so it's hydrophilic head end, or the polar phosphate head, which being polar means it will interact well with water. So the phosphate heads turn outward towards the watery environment of the cell and the environment that the cell is found in. The hydrophobic or fatty acid tails turn away from the water because they're hydrophobic or water repelling. They're fatty acids, right? So they're nonpolar. So they turn away. So this barrier that is the plasma membrane is a really, really good barrier because it really prevents most things from readily crossing from one side to the other. Basically, with all the processes that go on inside of a cell, each cell is going to require certain chemicals, certain substances to enter the cell, and they're going to require that certain waste products or production products leave the cell. And so this membrane provides a nice barrier that sort of helps to prevent mass movement in one direction or another. So we have the phospholipid. It's called the phospholipid bilayer because there's 2 layers of phospholipids. And we also have embedded proteins, and that's what those big purple blobs are on this screen. They are proteins. Those proteins are going to provide a variety of functions, which we'll talk about in a little minute. Now, if we want to talk about the phosphates, they're not glued together, right? They're only hydrogen bonded together because the polarity of the polar heads allow them to attract each other, but it's not like an impermeable barrier. So the phospholipids kind of move around. They move laterally, they flip-flop across, all the while maintaining that membrane. The membrane itself can have a couple of different characteristics that affect how fluid the membranes are. So some are very rigid and some are very, very fluid. And why does this happen? So we know about saturated and unsaturated fats. Well, that applies here as well. If the fatty acid tails of the plasma membrane phospholipids are unsaturated, they're going to be very flexible and very fluid. If those fatty acid tails are saturated, right, just like a saturated fat, they're going to be very rigid and static, and so that's going to be a lot thicker. So it's more fluid if it's unsaturated, it's more viscous or more solid if it's saturated. And that also calls into question the effect of temperature on the membrane. The warmer it is, right, the more fluid it should be, and the colder it is, the more solid it should be. That's where cholesterol comes into play. At the bottom right here, you see 4 cholesterol molecules, those 4 ring-structured lipids that are embedded in between the phospholipids, and that's sort of a more accurate picture of the membrane. So we have the phospholipids, the cholesterol, and we have the proteins. And the cholesterol provides stability of the membrane. When it gets really, really warm, it allows that the membrane still has some structure to it. And when it gets really, really cool, it allows that the membrane still has some fluidity to it. So the temperature and the cholesterol work together to sort of maintain the structure of the plasma membrane. So we have the phospholipids, we have the level of saturation, as well as the cholesterol and temperature affect how fluid the membrane is going to be. So when we take everything into consideration, this is a more accurate picture of what the plasma membrane actually looks like. You can still see the phospholipid bilayer, you can see the embedded cholesterol, all kinds of proteins. Then you'll also see some of those microfilaments on the inside of the cell in the orange, and you'll see fibers of the extracellular matrix on the outside. And basically this is all attachment points. So the organelles sit inside the cytoplasm. They are attached to the microfilaments, which attach to the plasma membrane. The plasma membrane then attaches to the extracellular matrix, and this allows that the cell sort of maintains its shape and location in the body by being attached at the inside and on the outside. So we have proteins, we have cholesterol, we have some carbohydrates there, and there's all kinds of different things that are going on at the membrane itself. So, which of the following best describes the structure of the membrane? So is it 2 layers of phospholipids with proteins embedded between them like a sandwich? Is it a mixture of covalently bonded phospholipids and proteins? Is it 2 layers of phospholipids with proteins crossing the layers or on the surface? Is it a fluid structure in which phospholipids and proteins move freely between each side? Or is it 2 layers of phospholipids with each layer covered with protein? It's the more accurate one. All right, so the answer here is going to be letter C. All right, the 2 layers of phospholipids with proteins either crossing the whole span or sitting on the surface, and that's what we see here, right? We see proteins, which would be integral proteins going all the way through. Sometimes we have peripheral proteins on one side or the other. So here is letter C. What about this? So if an animal cell will be more fluid at room temperature if it has what? More cholesterol, long-chain fatty acids, more unsaturated fat, or more saturated fat. So why is it going to be fluid? It's going to be fluid if it is warmer, which isn't an option. It's also going to be more fluid if it has what? Right? Unsaturated fat. So the answer here is C. More cholesterol would make it rigid. Longer chain fatty acids basically plays no role. Saturated fatty acids is going to be more rigid. All right, so the answer here is also C. The more unsaturated fatty acids there are in that membrane, the more fluid it's going to be. All right, so we know from the image that we just saw that there are all kinds of proteins that are found in the membrane or across the membrane or on the membrane. Now, these proteins are variable, right? And they're going to have variable roles that correspond with the roles of proteins that we talked about in chapter 3 when we did molecules of cells. So we talked about proteins being enzymes and signal transducers and transport proteins, right? Those aren't different, right? Those roles still find themselves in membrane proteins. And that's what this image shows here. Membrane proteins are created, right, like we said before, from the endoplasmic reticulum. And that's what you see on the top left. And then you see the little vesicles that sort of take those glycoproteins over. And they go to the Golgi, and some of them go out into sort of that aqua color, out of the cell. Some of them sort of circle back around and stay within the cell. But the endoplasmic reticulum, the rough endoplasmic reticulum, is where proteins are created. I would recommend that you go to mastering and take a look at this membrane transport video. It's excellent. It gives you a really good idea of what's going on inside the cell. And it really does sort of bring everything together before we discuss it. So what we're going to talk about now that we know about the structure of the membrane, one of the things that, again, the role of the membrane is going to be, or one of the roles of the membrane is to moderate what goes in and out of a cell. And The membrane itself is what's going to be called selectively permeable. Each cell type has a specific requirement, and so certain things can get in and out of, let's say, liver cells, for example, that may or may not be allowed in or out of skin cells. Right? Each cell is going to have its own membrane permeability properties. So some substances are going to be able to cross really easily, like oxygen and things like that. Some are going to take a little bit more effort. And so what we're going to do now is go through a variety of different transport mechanisms that substances can use to cross into and out of a cell or into and out of an organelle. Because remember, the membrane that surrounds the cell is similar in structure to the membranes that surround each of the membranous organelles, like mitochondria, ER, things like that. So this first slide is a slide of simple diffusion. It is a method of passive transport, and passive transport is basically the diffusion of particles using no energy. So diffusion is when particles separate out to equilibrium, right, using no energy. So on the top, you'll see there's a lot more yellow dye on the left. There's no yellow dye on the right when you start. And as we go through time, moving from left to right, by the time we get to the far right square, it's at what's called equilibrium. And that means the same number of particles on both sides of the membrane, an equal concentration on both sides of the membrane. The picture below just shows the diffusion of two solutes, meaning the yellow is going to go from left to right and the purple is going to go from right to left. But either way, when we get down to the end, bottom right, they're at equilibrium. The same number of molecules or particles on either side of that membrane. So that's called diffusion, simple diffusion. Things that move by simple diffusion are going to be oxygen, carbon dioxide, right? Kind of our basal, right? The most basic of molecules that we need to be able to cross the membrane relatively easily. So again, oxygen and carbon dioxide move by simple diffusion. Now, how do we know this, or what are the characteristics? Things that move really easily are going to be things that are very small and things that are non-polar or not charged. Those are the things that move really easily. So out of these, right, sodium is charged, glucose is huge and polar, H2PO4, that phosphate ion, is negatively charged. Water is polar. The only one on here that is non-polar is oxygen. Now, you may remember that we've said most things with oxygen are going to be polar, right? But that's only when it's pulling unequally on the shared electrons. If oxygen's pulling against oxygen, they are pulling equally against those electrons. So the answer to this would be B, because it is small and because it is nonpolar. Now we're going to talk about osmosis. Osmosis is another example of passive transport. It's basically the simple diffusion of water. Osmosis is the passive transport of water across a semi-permeable membrane, and usually that happens because the solute is unable to pass. The solute is the dissolved solid. We need to understand osmosis because it's very important in natural systems that water is balanced appropriately, right? We need the right amount of water on either side of the membrane. Doesn't matter, again, whether you're a tulip or a human being or whatever. Water balance is super important. So in this image, we have the two U-tubes at the top filled with water and some sort of solute. There's a membrane in between the two halves of the tube, and you'll notice that the spaces between the membrane segments are smaller than the solute is. So if we were looking at the top image, we would say that the purple molecules would move to the left, but since they can't do that, water moves to the right because we want to get to equilibrium. We want to get to an equal concentration of solute on both sides. The only way to do that is to move the water. And so osmosis is the movement of water, right, towards equilibrium. Now, this is why this is important. In an animal cell, we're going to learn three new words here: hypotonic, isotonic, and hypertonic. And you'll notice the colors in the squares: light blue, medium blue, and dark blue. That is supposed to indicate the solute level, the amount of dissolved solid in that water. So hypotonic means there's a low amount of dissolved solute, a lot of water, right? And when there's a lot of water, what's going to happen, right? So we can picture the animal cell has less concentration of water than the solution it's been placed in. So that is going to cause the water to go into the cell because remember, it's always trying to establish equilibrium. So hypotonic means more water, less solute. The water is going to go from high to low. If the water is higher outside and lower inside, it's going to go into that cell. And what happens to an animal cell? Boom. So animal cells do not like a hypotonic solution because a hypotonic solution, right, has more water and the animal cells will explode. Now plant cells, on the other hand, they really actually like a hypotonic solution because they want to take in as much water as they can. So a plant cell really likes that. In the middle, we have isotonic. Isotonic means there's equal solute levels on both sides of the membrane. And this is a red blood cell. An animal red blood cell really likes an isotonic solution. The amount of water that enters the cell is the same as the amount of water that leaves the cell. They're already at equilibrium, so the cell isn't going to shrivel up. It's not going to expand. It's just going to be able to metabolize and do its thing. So isotonic solution, equal concentration of water and solute on both sides, is great for an animal cell. Now, for a plant cell, they don't love that, right? It doesn't allow them to take in as much water as they need. They can live, but they're going to be wilty. That's what it means by flaccid, kind of wilty. On the right-hand side, we have a hypertonic solution. That's a high, let's say a high salt or high sugar. And that also means it's going to have a low amount of water. So if there's high water on the inside and low water on the outside, the water is going to leave the cell and the cell is going to shrivel up. We're always trying to establish equilibrium, right? Equilibrium is when the concentrations on both sides are equal. Now, cells might not like that, but that's what the chemistry is. It always strives for that. So the water is going to move high to low. The solutes, if they can, are going to move high to low. And we're always looking to, or the solutions are always looking to balance out. So let's take this situation. All right, so here we have a marine algae cell, right? Marine means salt water. So if it's a marine cell, it lives in salt water. All right, that means it has a high amount of salt, a low amount of water. If we take it out of the seawater and we put it into fresh water, what's going to happen? Right, so the initial concentration, if you think about it, the cell has a low volume of water, low concentration of water on the inside. If we put it into a high concentration of water, right, the water is going to what? Is the water going to leave the cell, stay the same, or is it going to increase in volume, right? And so basically we're looking at a situation on the left where we're taking a salty cell and we're putting it into fresh water. So it's going to absorb water and increase in volume. Some of these concepts are really difficult to understand, so please go back and forth, look at any of the videos, any of the extra videos. Let me know if you need a little training session on this, right? And I can help you out. But understanding iso, hypo, and hypertonic is going to be important. All right, another, or when we start talking about larger molecules or charged molecules or polar molecules, we have to figure out other ways to get them into and out of a cell, right? If they're not going to pass through simple diffusion, then we got to figure something out. So cells will also use a method called facilitated diffusion. And facilitated diffusion is a method by which a cell will use a protein, right, to cross the membrane. Facilitated diffusion, because it says diffusion, remember, that means it's going to be without energy. So just, if you want to think about it, it just makes a larger space for that molecule to pass. Now, what kinds of things pass into cells this way? They can be a little bit bigger. They can be a little bit maybe charged. Ions will move this way. Glucose will move this way, right, into the cell. And so we've got a couple of different things here. So we have, that's passive transport. Now, passive transport requires no energy, but there are transport mechanisms that do require energy, and that's what would be under the umbrella of active transport. Transport protein actively pumps a solute across the membrane against its concentration gradient. So when we're talking about passive transport, we're talking about things moving from a high concentration to a low concentration, sort of flowing downstream. However, when we're talking about active transport, we're actually going against that gradient, and we're going to take some, we're going to shove it into a place where there already is a lot. So when we look at this picture, you'll see at the top there are more Na plus or sodium circles than there are K plus, which are potassium circles. And the bottom, there are more potassium circles than there are sodium circles. So if this is active transport, it means that it's going to take the sodium from the bottom and push more of it out. It's going to take the potassium from the top and push more of it down. In some certain biological circumstances, sometimes you need to have a difference, right? Equilibrium might not be the best characteristic or property for the function. This is a image of what would be a neuron, where we're having an action potential or from the brain, right? A neural stimulus, and we need to have a difference between the amount of sodium on one side and the amount of potassium on the other side. So this is active transport. We're going to move things across the concentration gradient. This is called the sodium-potassium pump. All right, so you start at number 1. Sodium jumps in, and we push sodium out, and then it takes some potassium, and we push potassium in. It goes around and around and around and around, and it's always trying to create a differential between one side and the other, not striving for equilibrium. This is a great little summary. Diffusion, simple diffusion right through the membrane. Passive transport, no energy, using a protein. Active transport uses energy, and it moves the substances against their concentration gradient. Now, what happens if we want to move more than 1 or 2 little molecules, right? We want to move large quantities of things. Then we're talking about a process called exo and endocytosis, right? Exocytosis meaning releasing something, and endocytosis meaning pulling something in. But it's using the plasma membrane to engulf large quantities of things. So here we have 1 or 2 or 3, ions, molecules, small molecules passing across the membrane. If we're going to bring in a big giant food particle or a lot of water, right, we could do that using exo and endocytosis. Right, so all kinds of, we have passive transport, active transport, and then membrane-driven or exo and endocytosis. All right, so that is one section of this chapter, all right, in regards to getting substances to cross the membrane. The next thing we're going to do is a little bit of energy work. I'm going to talk a little bit about energy. So you can see from the picture up here, it looks like 2 squid, and the little blue dots are light sources, and they have the ability to produce these light sources through their metabolism. So we're going to talk about metabolism. We're going to do energy, kinetic and potential energy. We're going to do ATP and all kinds of different things. So when we talk about energy, right, there is sort of an overarching metabolism of the body or of an organism. And metabolism basically just means the number of, the sum total of the number of reactions that create energy and the sum total of the reactions that absorb energy or require or consume energy. So any of the chemical reactions that consume energy, build things up, pull energy in, are called anabolic. And any of the reactions that release energy, break things down, are called catabolic. So when you take your catabolism and your anabolism and you sort of add those together, you get overall body metabolism. And basically that's just a rearrangement of energy, how we take it in, how we rearrange it, and how we release it. It's sort of a capacity to cause change is what energy is. It is the rearranging of matter. That's how we see it. This picture here shows the potential and kinetic energies. Kinetic energy is the energy of movement, the energy of doing something, walking, jumping, breathing, flying. heat, light, those are all kinetic energy sources. Potential energy is the energy that something has sort of stored within it as a result of its location or its chemical arrangement. So something like gasoline, sugar, those are all potential energy sources. Potential energy is also seen in this image here of the diver being on top of the diving board, right? There's potential energy in regards to how far off the Earth's surface that object or person is. So potential energy is the energy that something has inherently as a result of its chemical arrangement or its location, which is height off the ground. And then kinetic energy is the energy of doing something. And energy can and is converted from one form to the other. So here we have a conversion of gasoline and oxygen in the car's energy to carbon dioxide and water, given off as heat. So we have the potential energy of gas given off in the kinetic energy of heat and movement. All right, on the bottom picture, we have glucose plus oxygen goes through the process of cellular respiration, giving rise to carbon dioxide and water. The high energy glucose is a potential energy source as it goes through the process of cellular respiration. Heat and cellular work result, right? Those are kinetic energies. So these are examples of a potential to kinetic energy conversion. All right, these are going to be exergonic reactions. Exergonic means energy is released. So these chemical reactions either store or release energy. This is two examples of potential energy sources of gas and glucose being converted into the kinetic energy sources of movement and heat. All right, so these are exergonic reactions. There are also endergonic reactions. An endergonic reaction is a reaction that requires energy input. These result in a lot of stored potential energy. For example, photosynthesis. Photosynthesis is the gathering up of the sun's kinetic energy, the kinetic light energy, and holding that into the potential energy of sugar. So we have the kinetic energy to potential energy conversion. It's endergonic. We're gathering it up and we're putting it into a nice, neat little sugar molecule. So when we think about the bottom image here of this exergonic reaction, you see ATP. ATP is actually a smaller form of chemical potential energy. It's called adenosine triphosphate. It is an adenosine molecule plus 3 phosphate ions attached to the end. And when you release one of those phosphate ions from ATP, you get what's called ADP. ATP is a nucleic acid, transfers energy from food to cellular work. And again, when the phosphate is removed, right, the ATP triphosphate becomes ADP and energy is released, right? So this is an energy releasing process, which means it is exergonic in nature. We're going to talk a lot about ATP in the next chapter. So what does ATP do for us? It helps with transport work, it helps with mechanical work, right? It's the little burst of sun energy. It looks like a sun. It's a little burst of energy that allows the cell to do what it needs to do. All right, this is another sort of way to represent the ATP-ADP energy cycle, right? So adenosine triphosphate plus water breaks. You'll see the energy released to the right? Energy released to go be used in cellular work. And then on the left-hand side, energy is brought in. So the ADP, so that left-hand side is an endergonic reaction, right? So we're taking energy in to create ATP. So we have the processes by which cells get and receive and remove substances, diffusion, osmosis, etc. We have energy, right? Chemical reactions require or release energy. All right, the next working cell topic is going to be enzymes. So enzymes are protein molecules. They serve it to catalyze reactions, either speed them up or they either make things. In this case, they break things down. Enzymes have a variety of different functions, Each enzyme is specific for what it does. So this is a sucrose molecule on the left, that disaccharide sugar. We are going to dehydrate it. We're going to hydrolyze it by adding water. We're going to add water to break that bond, but sucrase, the enzyme, helps to facilitate that more quickly. So enzymes are protein molecules that catalyze reactions. They don't contribute any energy, right? Many times they end in the suffix ase, which you can see here. Sucrase is a enzyme that digests sucrose. They are proteins, and their particular shape, right, is the reason for their selectivity. So this sucrase is a protein. It has a shape that fits sucrose. Sucrase is not going to fit lactose, right? Sucrase is not going to fit maltose, right? It only fits sucrose. So the reactant, sucrose is also called the substrate, is specific for that enzyme. Right? So it's important that we understand that enzymes are specific. They're proteins, and they do specific things inside the cell, build things up, break things down. So how do enzymes work, right? We have a reactant, and we want to get to the product. And the reactant without an enzyme on the left takes a lot more energy to make that happen. With an enzyme, right, it happens much more quickly. So enzymes are going to help to facilitate and catalyze reactions. All right, here is an enzyme in blue and the little tiny substrate molecule in red, and you'll notice how the shape fits right in that little nook, right? And when the substrate enters the active site of the enzyme, it grabs around it and holds on tight to it, right? So the enzyme starts with what's called an empty active site. The substrate enters the active site, the enzyme wraps around it, and then the enzyme is going to release its products. So here we have number one, enzyme, empty active site. The substrate enters that enzyme, it binds to it, water comes in, the substrate and the enzyme interact, and the enzyme breaks that bond with the help of the water. And here we have sucrose broken down by sucrase into glucose and fructose. So it's a breaking down. Now we also have other enzymes that build up. We have DNA polymerase. It's going to make a long chain of DNA. All right, so we're going to see enzymes all throughout the rest of the class. So understanding enzymes are proteins, enzymes are selective, and enzymes work to either create or break down molecules. Now, what else do we know about proteins? We know that protein structure is super important. We also talked about denaturing when we talked about molecules of protein in chapter 3. So this is not a different scenario, right? The environment of a cell affects enzyme activity, just like affecting the proteins from chapter 3. It's not different. Temperature, pH, salt concentration, those are all going to affect the shape of the protein. And each protein is going to have its own specific set of optimal conditions. So in this case, we have on the top, human enzymes work really well. Their optimal rate of reaction is highest, around 37. For heat-tolerant bacteria, right, the optimal is 77. At the bottom, pepsin, a stomach enzyme, works really well, right around pH 2. But trypsin, an intestinal enzyme, works really well at a pH of 8. So each environmental criteria affects the proteins in that environment, right? Because otherwise we can denature those proteins. All right, so this little guy, this is an enzyme catalyzed reaction. And how can the rate of the reaction be increased? So you'll notice as it goes from 0 to 2, 4, 6, 8, 10, 12, it starts to level off. How can we get that to go straight up, right? If we increase the concentration of the substrate, all we see is that it flattens out, starting at like 7, 8, 9, 10, forget about it. If we increase the amount of energy, that's not going to happen because we need to, it's the enzyme that's doing the catalyzing. So what we need to do here is increase the amount of the enzyme. If we increase the amount of the enzyme, we can increase the reaction rate because we'll have more enzymes doing more of their job. So the answer to this question is going to be B. One last thing about enzymes is that we want to make sure that we can have a method of controlling how often that they work. And that's when we're going to have an enzyme inhibitor. Enzyme inhibitors can determine how many molecules that the enzyme will react with. And we can have competitive or non-competitive inhibitors. This is a image here of a process of taking a molecule called threonine, and through a series of enzyme-driven steps, it ends up at a product called isoleucine. What you'll notice if you follow the red arrow is that isoleucine is going to actually bind with the enzyme and turn it off. This is a negative feedback reaction, right? Negative feedback reaction means that the product of a reaction actually slows down or stops the reaction. And when isoleucine is used up by the cell, the enzyme is turned back on again and makes more isoleucine. So this cycle will go around and around and around, turning on and off as the cell needs the product. It's one of the classic ways that cells can turn on and off production of certain cell products. So this is a negative feedback reaction where the product of the enzyme-driven reactions actually turns off the enzyme itself. Interesting stuff. So there's a lot here, right? I go through these PowerPoints. You guys aren't asking me any questions. I don't know where your questions lie. I kind of just go through the PowerPoints and sort of try to highlight some of the key concepts. But really, you need to be going in and looking at the different videos that I provide. You need to ask questions if you don't understand. You need to go to Pearson and use the study area. Use anything that you can find to try to firm up these concepts, because they can be a little, they can be challenging. So good job today. Thank you for listening. And I will see you for Chapter 6.